All right, so today we're going to talk about viruses and vaccines, and we're going to talk about COVID-19 a little bit more. And if we have a little bit of time at the end, we'll get into topic 3. I also have a Kahoot for you. That's going to be right here at the beginning. It's the same Kahoot I was going to give you last day. We'll get into that here in just a couple of minutes. So just a couple of things of review. We talked about viruses in terms of what they are. They are basically particles that have some sort of genome. That genome can be RNA or DNA, and that genome is surrounded by something called a capsid protein. And some viruses are also enveloped. Enveloped means they have basically a membrane, and embedded in that membrane are going to be some sort of glycoprotein that's part of the virus production. And in the case of SARS-CoV-2, this is the virus that causes COVID-19. We call those glycoproteins spike proteins, and that's what we're producing the vaccine against. So here's the picture of the SARS-CoV-2, which is the coronavirus that's giving us all the trouble these last couple of years. And like I said, I want to talk about COVID-19 in a minute here. Before we get there, though, I do want to do this Kahoot, and that'll help us review a few things about viruses. So let me just load that up here for you, and get that game pin going. You can load up the app, and we can get started here momentarily. So the game pin is 3349424. Okay, give you guys a few more seconds to join. If lots of people aren't going to do the Kahoots, I may discontinue them in the future. I had some good feedback from one of my other classes, but I know the whole online thing is a different experience, and it's a little bit easier to be passive. This was my goal, was to try to get people to be more active in the classes. So this one has 10 questions, by the way, and it's going to review a lot of concepts. Question 1, can viruses reproduce independently? So nice, easy one there. Everyone got that one correct. Well done. Although Ramil was a little faster. Question 2, what are the main components of a virus? Okay, so the correct answer is DNA or RNA. Some people selected the yellow orangish one. And you can see it says DNA and RNA. So remember, the genome is DNA or RNA. Question 3, what is the capsid around the viral genome made of? Gotta be quick. So capsid is made out of protein. Remember, that's what a virus is. It's basically a nucleic acid of some sort, and it's wrapped in a protein coat, and the protein coat is called the capsid. Number 4, viruses are classified by. Okay, everyone got that one correct. Well done. Viruses are classified by their genome type. We'll check on the scores in a minute. A specific virus can infect any organism. So false. Viruses tend to have a host range, right? So they can only infect humans, or they can only infect mammals or something like that. Maybe not the best worded question. All right, looks like we're neck and neck pretty much here. Number 6, which cycle is pictured here? I guess we didn't really get into this so much. So I'll tell you the answer, it's the lytic cycle. Although it looks like I have the incorrect answer selected. So the correct answer is lytic cycle, the blue one. I must have programmed this one incorrectly. I'll have to fix that one. Okay, question 7. What? Okay, I'll tell you the answer of this. The answer is provirus. We didn't talk about this. Sometimes things vary from semester to semester. This one also is HIV. We didn't talk about HIV this semester. All right, last question. Let's see if it's a tiebreaker here. True or false? COVID-19 is the virus and SARS-CoV-2 is the disease. False. Very good. COVID-19 is the disease and SARS-CoV-2 is the virus. So scoreboard. So Kyla, you held on to the lead and looks like you got the, oh, hold on, there's one more. Coronaviruses can cause. So correct. The answer is both mild and severe illnesses. All right, so Izzy with the bronze, and Emil with the silver, and I'm pretty sure we know Kyla came out on top. Well done, Kyla. All right. So I'll finish up there and we'll go back to our PowerPoints. What we want to talk about today is we want to talk about coronavirus disease and vaccines. So coronavirus disease is the long form of saying COVID, and 19 means it was discovered in 2019. So that's how they decided to name it. It's a little weird, but I guess it works when you have an acronym like that. So probably most people are kind of familiar with the symptoms of coronavirus disease, the most typical symptoms being things, respiratory symptoms, such as a cough or a sore throat or runny nose. Sometimes people have more severe symptoms, such as a shortness of breath or difficulty breathing. And you may notice I didn't mention the last two, fever and loss of sense of taste or smell. Those aren't usually due to the virus replicating. Those are actually due to their immune reactions. So they kind of fit into a different category. And I'll talk about that in a minute. And of course, one of the big crazy things about this virus is it seems to have, there's actually quite a number of symptoms. There have been people with, look up COVID toe if you're looking for something kind of weird. That seems to be a symptom in some cases as well. Myocarditis, inflammation of the heart is found in some people as well. So there's lots of weird things going on. One thing I wanted to do was just kind of compare COVID-19 with some other common respiratory infections. It's kind of worth mentioning them because there seems to be a lot of confusion. You know, people calling COVID-19 the flu. Well, it's not. It's actually a different disease. And so one way to compare these things, I have COVID up there, influenza, and the common cold. And just to show you that they're actually different viruses. COVID-19 is caused by a SARS coronavirus. Influenza is caused by influenza viruses. The common cold is actually a whole bunch of different viruses. The main one we've kind of mentioned once or twice here is something called rhinoviruses. There's also some coronaviruses that cause colds and adenoviruses and a couple of other things like parainfluenza and whatnot. But they are different viruses. So that's one thing that's kind of worth it to know. In terms of prevention, we can even talk about things like vaccines. There are no vaccines for the common cold. We just get it and we just suffer, and that's kind of it. Usually we don't suffer too much. Cold is not such a bad disease to have. Usually goes away after a day or two. Influenza, there's an annual flu shot. You may have heard about that kind of thing. It mutates. Influenza mutates pretty fast, enough that there are different strains every year that they have to reformulate the entire vaccine annually. COVID-19 does mutate, not as quickly. We are at the stage where we're ending up with different variants, but the vaccines we have developed, of which there are 4 available in Canada, are all are all still effective to a certain degree with the new variants. Maybe someday we'll have to reformulate those vaccines. Another way to look at it is the symptoms, and you can see they all have kind of overlapping symptoms, right? COVID-19, some of the common symptoms are fever and cough. Sometimes people have headaches, sometimes people lose their sense of smell. That's kind of a weird one. Losing your sense of smell and taste doesn't happen with a lot of other illnesses. So that's kind of one that happens in about a quarter of infections. It's not on this list because it's not common to the other diseases. Influenza, very common, have a fever. Much more common have aches and pains and extreme fatigue with influenza. Common cold is a little bit more runny nose. Rhinoviruses mostly infect the nose and sinuses, so the symptoms are kind of mostly kind of stuck to that part of the body. Sore throat, runny nose, sometimes a headache, pretty rare to have a headache with a cold, but sore throat and sneezing and nose kind of symptoms are more common with the common cold. So this is one way to kind of think about these diseases. They do have overlapping symptoms. They are different viruses and they affect the body differently. I guess I had started circling them, and there's the boxes around them. So something that I had kind of alluded to in terms of how does a virus actually cause the disease? Because what we talked about last week was one thing was that viruses, many of them have no effect on human health. There's kind of two things going on really in terms of how the virus causes disease. Number one, the virus kills cells. So this is a respiratory virus, and so it's killing respiratory cells down the trachea, down into the lungs, and depending on how deep it gets into the body, it might be a sore throat, which is kind of upper respiratory system. If it gets deeper, you could be coughing or having pain, in the chest, those kind of things. So that's kind of number one, right? Virus kills cells in that body system, leads to symptoms in that body system. The other thing is basically immune responses of different sorts. So the direct immune responses include things like fever, fatigue, and chills. Those are all kind of standard symptoms that people see with all sorts of different infections because it's the immune system kind of ramping up, and it's a consequence of that. Many of the other symptoms with COVID-19 are due to sort of longer-term inflammation. So the SARS viruses, it turns out that they are not just respiratory viruses, but they actually do infect the vascular system. So sometimes people end up with all sorts of really bad inflammation in different parts of the body. I mentioned the myocarditis, that's inflammation of the heart. Sometimes people end up with long-term inflammation in the lungs, which is obviously very serious. And all sorts of weird symptoms in another part of the body, the loss of taste, the COVID toe, those kind of things. So like I said, you can kind of think of it as two prongs. Number one, the virus kills cells. That leads to damage in the body systems. Number two, the immune system is doing something, whether it's longer term or shorter term. And this is why, one reason why vaccination is such a good idea, because we can prevent a lot of these long-term issues. Makes the disease a lot less severe in most cases. This is just another graphic I found on the internet kind of showing how the virus is attacking cells in the respiratory system. I don't want you to get bogged down in the details, but you can see the lungs there and you can get the idea. You can always take a look at it anytime that you like, if you download the PowerPoint. Okay, so I wanted to switch and gears and talk about immunity in vaccines today. So this is a slide kind of giving you a simplified view of the immune response to a viral infection. So if you take a look up at the very top there, you can see there's the virus, and the virus there is being recognized right here. This is an immune cell here. You can call it a white blood cell. There's all sorts of different names of these white blood cells, but we'll just call it an immune cell. Immune cell. So that immune cell has, it's recognized the virus, and what it does is it destroys it. But one of the things that it does, which is kind of weird, but I kind of think of it as like a guard dog. In one of those movies, you may imagine a guard dog, they're running around a building or a property, somebody gets in and they chase them and hunt them down. And in the movies they might have a little piece of their clothing or something after the burglar has escaped over the fence or something like that. This is kind of what these immune cells do: is they hold on to a piece, and you can see right here it's holding on to a piece of the virus. And this is something that gets recognized by other immune cells, and there's a whole bunch of different varieties of immune cells. There's B cells and T cells. I'm not going to get into all the specifics on those. But what happens is these cells start to produce something called antibodies. And antibodies are proteins, they're shown right here. And these proteins will recognize that virus in the future. So these B cells and T cells, they're long-lived. They can be long-lived memory cells. And so next time you end up with the infection, if you produce enough and the right types of antibodies, then these antibodies are going to bind the virus like this and mark it for destruction. So that's kind of a really quick whirlwind tour through the immune system in terms of how it's recognizing a future infection. And so hopefully the idea is if you ever encounter that virus again, the disease will be much less severe or maybe not even existent because our immune system is recognizing it and attacking it. So notice I said it's not preventing infection. This is one of the misconceptions around vaccines, is that vaccines don't prevent infection. They minimize disease and make it either non-existent or less severe. But most vaccines don't actually prevent infection. They're kind of like more like a fire extinguisher, right? The fire comes and then they put it out, right? So I want to spend a few minutes here kind of talking about some of the different types of vaccines that are out there, and in terms of how the technologies work, and talk a little bit about the COVID-19 vaccines. Here's some of the strategies here, and I'll go through all of them, and pretty much every single vaccine strategy has been used by researchers around the world. And they have, there's different types of vaccines found in different countries that are for COVID-19 that are using some of these different strategies. In Canada, we have basically two types of vaccines we're using, and partly because they were developed quicker and seem to be safer than some of the other methods, at least for this disease. So let's just talk about kind of traditional vaccines. So traditional vaccines fit into two categories. One is attenuated. So attenuated means a weakened pathogen. Weakened pathogen. And the other group is inactivated. Inactivated usually means you're killing it somehow. So let's talk about these. So the whole idea here is you have the pathogen, and we want to make it so it's not dangerous anymore. So you can kill it. You can kill it using chemicals. Sometimes they use UV radiation. There's a number of methods, but typically it's chemicals such as formaldehyde or something like that. And so now you have the pathogen, but it's no longer dangerous because it can't replicate in the human. Attenuated is a weakened version. So there's different ways to make weakened versions of the same pathogen. And so you can breed them, and there's genetic strategies for doing so. I'm not going to get into all that, but same idea is you have a pathogen now that's not going to hopefully do any harm. It's still alive, but if it's done right, it's not going to be dangerous, but it still is the virus. And so you can put that in the body, either the killed or the attenuated form, and then the body is going to recognize it as a foreign invader. It's going to make some of those immune cells, those long-lived memory cells. And so when there's a secondary infection down the road, it's going to recognize that virus and hopefully destroy it and prevent disease. So those are kind of the traditional strategies for making vaccines. If you go back, we're talking about hundreds of years now, right? Vaccines are technologies that have been around for like 400 years, and they've with some great success for a lot of infectious diseases. And I'll show you some data here in a couple of minutes. So this is the traditional technology. Like I said, this is representing two types of vaccines, killed and weakened, which are basically using the pathogen itself. There's a lot of newer vaccines. for COVID, we're not using this type in Canada, but for other vaccines in Canada, such as you may have as a child gotten I'm thinking about the meningitis vaccine, the Hpv vaccine. These are all subunit conjugate vaccines. I'm not going to get into the differences between the subunit and conjugate, but basically what you're doing is rather than injecting the entire pathogen, you're just finding one part of it, right? That's why it's called a subunit vaccine. So you take that component, and that component on its own is not going to be dangerous to the human. And so in the case of COVID-19, it's usually that spike protein is what we're targeting because it's on the surface of the virus. So what you do is that's part of the formulation. It gets part of the vaccine injected into the person, and they have an immune response, in this case against the spike protein or just that component. So when the actual virus shows up, part of it is recognized, and then the immune system can mount a response against it. And works quite effective for some of these vaccines. The pneumonia and meningitis vaccines have greatly decreased those diseases in children in Canada in the past 20 years by quite a bit. And I have some numbers here to show you here in a minute or two. So let's talk about these new vaccines, because the COVID-19 vaccines in Canada are using some new technology. And I think it's pretty cool. There's DNA and RNA versions of these vaccines. At the moment, there's no DNA vaccines that are being used in any form in Canada. So I'm just going to talk about the RNA vaccines, and sometimes we call them messenger RNA vaccines, right? mRNA. And we're going to talk a lot about messenger RNA in the last part of the course. But basically the way it works is you have some RNA, and RNA, of course, is genetic instructions. That's it. So you're injecting RNA, and usually these are coated with a special type of lipid, which I'm not sure what it exactly which lipids it's used. But the whole idea is that you're injecting RNA, genetic instructions, into somebody's tissues. And the lipids are used there to kind of help that RNA get into cells. So if you had this vaccine, it's probably got injected into your arm, into the muscle tissue, and the muscle tissue has cells, right? And so what happens is that RNA, here's the RNA right here, it gets into the cells, and it finds a ribosome. And so a ribosome is designed to take that messenger RNA and make amino acids out of it. So it strings together the amino acids, and those amino acids, in this case, for the coronavirus vaccine, they code for that spike protein. So rather than injecting the spike protein directly, we're injecting the instructions to make that spike protein. And so the spike protein then gets into the blood, and you have your immune response, and that's the whole idea. So in this case, rather than, it's kind of like, picture nowadays we have the International Space Station, right? And let's say they need a tool. Let's say they didn't need a new type of wrench. Well, we could fly the space shuttle up there and bring the tools in. But nowadays they have a 3D printer up there. So rather than sending up the tool all by itself, it's just send up the instructions. And that's what we're doing in this case here. It's kind of like sending those some instructions over, our cells instructions like an email, and they make that viral protein. And RNA is a very short-lived molecule. And so the RNA is not going to linger around and have any effects at all. And it just kind of disintegrates inside the cell. But you still get the immune response. And these vaccines are super easy to manufacture, it turns out, which is why they kind of, were the leaders in this vaccine race, because you don't need to, in this case, grow any viruses. Growing viruses is dangerous stuff. You need level 3 labs, and you need highly trained personnel. In this case, no virus particles actually required. All you need is a genetic sequence, and it took them only 4 days to design this thing. So pretty incredible. Now, 4 days basically represented 20 years of research before we could get to that point. And we were very lucky that we were at this point in this technology that it was ready to go for this pandemic, because like I said, it represented about 20 years of work for a lot of people to be able to get to this point. So there's one last type of vaccine that is also new, that is relevant to COVID-19, and these are these viral vector vaccines. So this is a little bit different as well. So I mentioned that in this previous slide, right, these DNA vaccines, there's nothing for use in humans, so they're kind of not worth talking about. But there is a way to get DNA in, and we can use what is called a viral vector. And so this is the same idea as the messenger RNA vaccines, except for this time we're delivering DNA. How we're doing it is we're using a repurposed virus. So in this case here, you can see, for example, you can re-engineer a measles virus or re-engineer an adenovirus. And so viruses are designed to basically deliver genetic material to cells. That's what they do. And usually those instructions say, make more viruses. In this case, what we've done is we've repurposed these viruses, and they're only delivering the instructions to make spike proteins. So same idea as the messenger RNA, we're just getting the DNA in the cell in a different way. And the instructions say make spike proteins. Our cells make the spike proteins, and those spike proteins lead to an immune response, which is hopefully going to give you protection against disease. So pretty cool. Vaccines are pretty cool technology, and I think a lot of people don't really understand what they are. And there's a lot of fear and misunderstanding around vaccines, which kind of I find really sad, because it's I think honestly vaccines are really incredible technology. So as of now, there's 4 vaccines for COVID-19 that are approved in Canada. So the first 2 here, you probably know them by their company name, Pfizer and Moderna. Those are messenger RNA vaccines. You can see they have trade names. Comirnaty is the Pfizer vaccine, and Spikevax is the Moderna one. So kind of some fancy names. These ones here are approved for different profiles. So the Pfizer vaccine was recently approved a month or 2 ago for ages 5 and up, and the Moderna vaccine for ages 12. The other 2 types of vaccines approved in Canada are the AstraZeneca vaccine, which is also the trade name is what is it, Vaxzevria. So again, they have to have a name for it, I guess, and it's approved for 18 and up. And the one that we haven't heard of as much, actually, I don't even know anybody who's had the Johnson & Johnson vaccine, but it's also a viral vector vaccine. And so at the moment, the most effective vaccines actually seem to be the Pfizer and Moderna vaccines, which seems to be the case. And also they seem to be slightly safer than the other ones. And I'll talk about side effects here in a moment. Okay, so I do have some questions and answers here. And if you have any additional questions once I'm done this, please throw them into the chat box or speak up. But I have some questions and answers about these. And these are things that type of questions I get a lot of people asking about, or there's a lot of misconceptions around on the internet. So the first question is, did they skip animal testing or other steps? The answer is no. As I mentioned it, these vaccines were 20 years in the making in terms of the research and all that. And actually, both of these types of vaccines, the messenger RNA and the viral vector vaccines, had been both designed initially for Ebola and a few other purposes. And it's very easy to put a new genetic sequence in there. And so January of 2020, once the new virus had been discovered and people were concerned about it becoming a pandemic virus, they started designing them and were able to do so in a short amount of time. The reason why things were so fast is because of the amount of money pumped into this kind of thing. If you think about a typical kind of drug or something that's made and all the red tape that has to go through these things, your typical drug, usually what happens is you do your animal testing, and usually you do it on let's say a dozen rats, and then you take 6 months to analyze the data and apply for the paperwork for the next steps, and then maybe you do it on a larger number of rats, and then all the paperwork goes through, and the government takes 6 months to approve the human trials and those kind of things, and every step gets more and more expensive. So that's why they take so much time, because of the risk. Doing a dozen rats isn't too expensive. Doing a few hundred rats is more expensive. Any human testing gets really, really expensive. So there's all that risk that the company is doing. That's why they do it slowly, because they want to pull the plug on anything that doesn't work. In this case, with these vaccines, governments around the world were pumping billions of dollars into this. And so the other thing they were doing was speeding things up in terms of not sitting on the paperwork for 6 months. So what that means is that you can have your human trial not necessarily approved before the animal testing is done, but have it ready to go for basically the next day, so that you can do your animal testing, have the paperwork submitted, and the government is, they're looking at it, they're taking a very short amount of time to look at it, and the human testing is approved and ready to go. And so money basically speeds things up, is what's going on. In Canada, we have a very stringent, strict process, probably one of the most strict processes in the entire world, in terms of approving drugs and new procedures. And I have a lot of confidence in the system, quite honestly. Another question that people ask is are these nucleic acid vaccines going to make me into some sort of weird mutant? No, it's not exactly how RNA works. RNA is a short-lived, fragile molecule. It's not going to mess around. It's not going to incorporate itself into your genome. That's science fiction. So what about the effectiveness? What does that mean? Because there's a lot of numbers thrown around, and I can sort of give you the numbers for the original trial. And every study, you got to look at the numbers and see where they come from. And usually what they do with these studies is they're looking at a large number of people. You can see in the Pfizer vaccine there, they were actually testing more than 40,000 people. And so in that case, they literally had two groups. Half had no vaccine, half had a placebo. A placebo is where a person thinks they're getting the vaccine. Maybe they get a shot of saline instead, or a different vaccine for, say, measles or something like that. And then they watch to see who's going to get sick and how many, right? And so in this trial of more than 40,000 people, 170 cases of COVID were observed, of which most of those cases, 162, were in the placebo group versus the vaccine group. So if you look at that, 162 divided by 170 is basically 95%. So that's where those numbers come from. Looking at the newer variants and things like that, the math and the statistics gets a little bit more complex. But that's kind of basically what they're doing, is they're looking at the number of cases, and then they're looking at vaccine status and which vaccine you had and which variant, and it's mostly just statistics. But that's pretty good, actually. 95%. A lot of vaccines are 80, 85%. Some are as low as 60% effective. So this is 95 is actually a very, very good standard. So immunity, this is something that we're still learning quite a bit about. Immunity studies can take years, and we're learning a lot about this. A lot of people are asking, what if I have COVID-19? Am I naturally immune? You will have some immunity if you've had COVID-19. The question is how much and how good is it? Right now, some countries are basically saying, okay, if you've had COVID-19, we're going to consider you immune for 90 days. The research seems to show that immunity from the vaccines does seem to be better than having immunity from the infection. There's a lot of misconceptions on the internet, people saying that natural immunity is good enough, and it might be in some people, but the data is showing otherwise, that the vaccine immunity is better. Quite honestly, the best immunity is if you've had both. That's what the data is really showing, that if you've had COVID-19 and 2 shots of vaccine, then you're having good immunity. And these are the kind of things that the recommendations change. And there's a lot of debate about getting the booster shots and those kind of things. And this is the kind of thing where the jury is out. There really have not been a lot of studies on the boosters and whatnot. There's been a couple that shows that it does seem to help with immunity against the Omicron variant and things like that. But the long-term advice may change on that. The reality is this may be a 3-shot vaccine. There are other 3-shot vaccines out there. A lot of people don't seem to know that. Hepatitis B vaccine, at least originally, used to be a 3-shot vaccine. I don't know if it still is, and so on. I guess we'll see. Time will tell. There's the other question about immunity to these things in terms of we don't really understand immunity to coronaviruses that well. And it does seem that immunity does wane. Some viruses' immunity will last for years. Coronaviruses, it's looking that maybe after 6 months, immunity does go down. It doesn't mean that you're going to get sick. And that's really the end point, I think, of this this pandemic is really trying to, prevent people from getting terribly ill. In the end, we may have to live with this thing and just get kind of, colds from it once in a while. And hopefully that's the worst once everyone has enough immunity to it through vaccination or natural immunity. So like I said, immunity seems to last at least 6 months, but there seems to be some evidence that it does start to wane, at least in some people. We just haven't had the vaccines around long enough to fully have a good answer to this. And the good news is that some people do seem to get reinfected, but the good news is that in most cases, if they've been vaccinated, the disease is not serious, even if they do get an infection after having the vaccine. And that's kind of the goal, right, is to prevent deaths and severe illness. That's really what we want to see from this whole thing. Like I said before, vaccines don't necessarily prevent infection, but they usually prevent disease, or at least make the disease a lot less severe. And that's what we want in the end, is to have a lot less severe disease. What about side effects? Let's talk about side effects. If you look up side effects on the internet and go to official sources, these are usually the ones that we're looking at getting. Most of them are kind of obvious. You're getting pain at the injection site because you just punctured your skin and some tissues. That's the most common side effect people get. And a lot of people get other minor side effects. When I had my vaccine, I had a headache the next day, and that was the worst of it. And I was tired, and I don't know, it could have just been I was tired, but I think it was a vaccine side effect. Most of these side effects here, headache, fever, fatigue, chills, these are actually the immune system acting. Remember, I mentioned that that's part of how we get viral symptoms. In most cases, these are much less severe than what you would have with the actual virus. People who get a fever with a vaccine is usually quite mild and good enough to just treat with the Tylenol. Fever with illness, often people are bedridden and whatnot. There are some rare side effects. I can't remember, I have a slide on this as well. There are some rare side effects that have been in the news around a vaccine, such as myocarditis. There was the blood clots with AstraZeneca and so on. And I could talk a lot about those side effects, but I will say just a couple of things about them. One is that yes, there are side effects like that. They tend to be quite rare, maybe 1 in 100,000, 1 in half a million kind of thing. And all those side effects are more common with the actual viral infection. If you look up the myocarditis, that's inflammation of the heart, people who've had the infection are more likely to have it than people who've had the vaccine. And this is part of the immune system interacting with that antigen. I'm not going to get into all those in too much more detail, other than that they're quite rare and in most cases not serious, and they're overblown by the media. And those are my opinions on that. Some people ask about other conditions, allergies, and other immunological conditions. Those are the kind of things that you should be talking to your doctor about. I'm not going to comment on that, other than there are some reasons for people to avoid the vaccines for certain types of allergies and other conditions. I guess I talked about some of these serious side effects. I'm going to kind of just move on from that. Another one is Bell's palsy. You can look that one up. Again, it's more common with the viral infection than the actual vaccine. That's kind of it for my questions and answers. If anyone has any specific questions, just throw them in the chat, or you can talk to me about it later. Just some more thoughts and opinions on this thing, and of which I have many. As you may have guessed, I really do believe vaccines are amazing technologies. And I'll give you a little bit of data here in a moment to kind of back that up And I hope that you do too, and that you can share some of this information with the people that you know. I think one of the biggest issues and threats to public health in the last 20 years has been vaccination hesitancy. And look at where we're at, right, with this pandemic, right, in terms of all the misinformation that's found out there, and you know, people are making irrational decisions around this, and I just find this whole thing quite heartbreaking. I do think the technologies are super cool, and if you have any questions, I'd love to talk to you about them anytime. I was going to show you a slide here kind of showing some statistics I mentioned on vaccines. And this is, if you take a look at the top part of the slide, it's showing statistics for older vaccines, so available before 1980. And you can see this is the number of cases in the US, but it's very similar percentages in Canada. And so you can see, like, for example, diphtheria is the first one on the list. Basically, cases of diphtheria are at 0. I have never heard of anyone in my lifetime getting diphtheria. My great-grandmother died of diphtheria, but that is the only time I've ever heard of anyone getting diphtheria, and that was a long time ago. A lot of these other diseases, measles, mumps, polio, rubella, you know, those kind of things, I've met people who've had them, and most of them were like people who are you know, in their 60s. They had them as kids before the vaccines were available. So a lot of these diseases have plummeted to near 0, to the point where none of us have probably ever seen any of them in our lifetime, which is really incredible. There's some newer vaccines. You can see different rates of success here: hepatitis A, hepatitis B, varicella. This is chickenpox, of course, and the amount of disease prevalence of these things has plummeted. And in many cases, most of these mean reduced amounts of deaths in children and other people. And I really think vaccines are very, very incredible. So that is kind of the end of that. So hopefully you found some useful information in there. Most of that has been information, a few opinions in there, but I think my opinions are founded by science, and I think they're accurate. I've tried to make it clear when I was not sure about the information, but if you do have any further questions on these things, do let me know. I know a lot about this stuff, and I love to talk about it a lot. Looks like that's it for the virus topic and vaccines. And so just a little heads up on this. I did have on the last semester, on one of the midterms, I did have a matching questions about the different types of vaccines. So I do expect you to know a little bit about the different types of vaccines, enough to do a matching question or a multiple choice or 2 or something like that. All right, I'm going to not finished yet today. What I want to do is maybe just start and do a quick introduction into the next topic, and because it is related to our lab, and we are having our lab tomorrow. So let me just pull that up. So topic 3 is about microscopy and a few other types of laboratory techniques. And the main thing that I wanted to show you today, sorry, I know I just minimized that. I gotta pull it back up. Main thing I wanted to show you today was a short video, and a short video on microscope usage. And that's kind of the only thing I wanted to show you today, because it might help you in tomorrow's lab. So let me play that for you. It's very short, and I'll make a couple of comments about tomorrow's lab, and then that will be it for today. So I will play that for you right now.
In this video, I'm going to demonstrate how to focus the microscope. Begin by turning your microscope on and adjusting the light intensity to about 5 or 6. Then place your slide in the clamp on the stage and center the specimen over the condenser. Then turn the low power objective into place and use the coarse focus to move the stage all the way to the top. While looking through the objectives, slowly turn the coarse focus down until you can find your specimen. When you think you've found it, wiggle the stage a little bit to make sure what you're focusing on actually moves with the slide. If you wiggle the stage and nothing moves, you're focused on the wrong plane. Once you've focused at 10x, you're going to rotate the next power into place, the 40x. This time, use only the fine focus to adjust the stage and focus on your specimen. You should not need to turn this focus more than one complete rotation. Once you've focused on your specimen at 40x, now you're ready to move up to oil immersion. Turn the 40x objective out of the way and put a drop of immersion oil over your specimen. And then rotate the 100x objective into position. And it should touch the drop of oil. Once you have the 100x in position, use the fine focus to focus on your specimen at 100x. When you're done, be careful to turn the objective out of the way before you lower the stage. Clean your microscope and put it away.
Okay, so I just wanted to show you that video. I probably should have shown it to you last week. I kind of forgot about it. But just some more tips for using microscopes, because we are going to use the microscopes again in tomorrow's lab. So I'll talk more about tomorrow's lab tomorrow afternoon. When you show up, do take a quick look at the procedure and whatnot to see what you're going to be doing. And I'll cover the details tomorrow. And other than that, I guess that is all for today. I'll see everyone tomorrow afternoon, 2 o'clock at the biology lab. So take care.